morning, good afternoon. Thank you all for joining us today. We're going to let everyone join in and get settled for just a quick moment, and we'll get started briefly. All right. Well, hello, everyone, and welcome to today's webinar titled Unleashing the Power of the Immune System. Live cell analysis provides key data for cancer research. This webinar is a part of the 11th annual event in the Cancer Research and Immuno-oncology Week virtual event series. I am Kaylee Bach of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and sponsored by Sartorius. For more information about Sartorius, please go to www.sartorius.com. Before we begin, I would like to remind everyone that this event is interactive. We encourage you first to participate by communicating with other attendees using our live chat feature during the presentation. You can find the live chat located at the left of your screen. You can also participate by submitting as many questions as you would like during the presentation. To do so, simply type them into the Ask a Question box and click Submit. We'll answer as many questions as we have time for at the end of the presentation. If you have trouble seeing or hearing the presentation, click on the Help Desk button located at the top of your screen within the event. All right, now let's get started. I'd like to now present today's speaker, Nicola Bevan, Manager, Cell Imaging Applications at Sartorius. For a complete biography on our speaker, please find it on the left of your screen. Nicola, you may now begin your presentation.
Hello, I'm Nicola Bevan, and I'm part of the BioAnalytics team involved in the development of new products and applications for use on the Incucyte Live-Cell Analysis System. I work for Sartorius, based in their UK office near Cambridge, and I am here to talk to you today about unleashing the power of the immune system and how live cell analysis can provide key data to support cancer research. In today's presentation, I will first introduce the concept of live cell analysis and the Incucyte system. I will then demonstrate how it can be used in the functional assessment of CAR T cells with the use of an immune cell killing assay. I will then move on to demonstrate the Incucyte's applicability in the area of functional assessment of antibodies before concluding my presentation. So to set the scene, there is a general interest to deliver more translational cancer cell models in the aim to provide more relevant and predictive in vitro assays. This change in model design is driven by both the use of increased cell relevance, moving away from immortalized cell lines towards patient-specific cells, and also model organization, moving from traditional 2D monoculture towards 3D models, microtissues, and organs on a chip. The data I will share today will mainly use co-culture models using both tumor and immune cells. The use of 3D spheroid and organoid assays are a step towards this increased complexity and are becoming more popular and easier to use in the in vitro setting. This increase in model complexity also drives the requirement for enhanced data analysis to capture all the information from these models. The cellular models can be enhanced further to incorporate patient-derived material or the inclusion of different cell types. So let me introduce the Incucyte system. The Incucyte Live-Cell Analysis System is designed to efficiently capture cellular changes where they happen inside the incubator. It has multiple trays to facilitate the use of various cell culture vessels, enabling up to 696 or 384 well plates to be imaged at one time. It captures high-resolution fluorescence and phase contrast images with the use of ejectives that move under the culture vessels so the cells remain static. It is equipped with a 4x, 10x and 20x ejective, which can be chosen in the software to match the biology of interest. It will then record data and quantify in real time over hours, days or weeks, controlled by user-defined time-lapse intervals. This flexible system enables users to observe, quantify complex biological changes in real time. An integrated software simplifies the data analysis to speed time to answer while producing publication quality graphs and plots. So what is live cell analysis? This is the continuous measurement of cellular functions, for example, growth, movement, and reaction to drugs. The monitoring of cells is applicable across the entire experimental timeframe, from initial cell seeding in a flask or plate to manipulation of cells, for example, transfection or differentiation, to finally the assay phase in response to a drug. The image is captured and analysis happens in real time, and as the cells are maintained within the incubator, they are kept at an optimal temperature, humidity and environment for reduced cell perturbance. The assays can be left to run as long as required, from hours to months, depending on the biology being captured. We offer an overall solution, including hardware, software, reagents and applications, to enable our customers to maximise their biological insights. The Incucyte is shown here inside the incubator with the controller unit sitting on top, and this is the computing power of the system which controls the instrument and where the images are stored and analysed. The system can be accessed from any desktop networked to the system, so users can remotely control the image acquisition, review images, analyse experiments and interact with the results. We offer multiple software modules which enable different image acquisition and analysis options, alongside optimised reagents for long-term live cell assays. The image taken from our website shows the various applications we have worked on, showcasing example data sets and easy access to multiple collateral items which can be downloaded from protocols to white papers. Let's now consider our biological story, and I'll start by introducing you all to the immune cell killing assay. Researchers have been investigating this immune response for many years, with the potential of embracing the immune's ability to fight cancer. More recently, with the rise of cell-based therapies like CAR-T therapy and monoclonal antibody approaches, this biological response has grown in importance. The diagram highlights many of the immune cells, like natural killer and T cells, that can contribute to this response, and some of the key processes and markers that are involved in the immune and tumour cell interface. There are also a multitude of methods available to help interrogate this response, but as shown here, some have limitations, such as the use of radioactivity, endpoint analysis or complicated methods. Our live cell analysis approach offers an alternative, which is a kinetic image-based assessment of the biological event in real time, which can be easily quantified. This slide highlights the standard method used for the Incucyte assay. Target tumour cells are first seeded into flat-bottom 96 or 384 well plates and left to adhere to the plastic. The assay can be set up using either adherent or non-adherent target cells, but we do advise that these are labelled either stably or transiently with a fluorescent cell marker, for example Incucyte Cytolite Rapid Dye. This then simplifies target cell identification for easy quantification. Additions can then be made to the target cells, for example addition of immune cell activators, cell health reagents or test antibodies. Lastly, the immune cells of choice are added to the well at the desired target to effector cell ratio. Plates are then placed in the Incucyte and images collected. We recommend either using a 10x or a 20x objective depending on the choice of target cells. The video shows this assay in action. This well has SCOV3 cells expressing a nuclear-restricted red fluorescent protein in the presence of freshly isolated PBMCs. These are a target to effector cell ratio of 1 to 10, and they are also in the presence of anti-CD3 antibody and IL-2 to activate the T cell population, and caspase-3-7 green to track cell apoptosis. If you watch the cell in the yellow ring, you will see that the T cells interact with the tumour cells on multiple occasions before the cell enters degranulation and ultimately death, indicated by the loss of the red fluorescence and an increase in the green fluorescence from the caspase-3-7 reagent. Using the Incucyte, we can then quantify the loss of the red fluorescence, which is indicating direct tumour cell death, and the gain of green fluorescence indicating target cell death over time. We can use both Incucyte software tools to also capture, we can use other Incucyte software tools, to capture details from the effector cell population if this is of interest. I'm now going to demonstrate how this assay has been used to study the killing with CAR T cells. As everyone is aware, cell therapy is a fast expanding area of immunotherapy, offering a personalised, intelligent medicine with potential for disease cure. Chimeric antigen receptor or artificial T-cell receptor therapy is expanding rapidly with impressive clinical results, especially in the area of B-cell lymphoma treatment, with a recent paper detailing 10 years remission in 2 chronic lymphocytic leukemic patients. The CAR-Ts are designed to recognise target cells expressing a specific surface marker, which enables enhanced antigen-specific tumour clearance. Our live-cell analysis assay, in combination with high-throughput flow cytometry, can add value during CAR-T development by first confirming biological effectiveness and target epitope interactions, but also during expansion of CAR-T cells to assess that appropriate phenotypes are being conserved. The video, taken over 3 days, shown on the left, anti-CD19 CAR-T cells supplied by Professor Rafiq's lab in UCL are attacking antigen-positive NALM6 green cells. Notice how the immune cells flock around the target cells before killing them. They also show a change in morphology, becoming larger and less spherical before showing signs of proliferation. The right-hand video shows non-transduced cells having no effect on the growth of the target cells. The example shown here is also using anti-CD19 CAR-T cells, but this time commercially purchased as a mixed population with around a 50% transduction efficiency for the CAR construct. We tested them at various target to effector cell ratios against antigen-positive Ramos cells or antigen-negative Urcat cells, both expressing a green fluorescent marker for easy quantification and tracking. As can be seen in the images, the anti-CD19 CAR-T cells have herded the Ramos cells into clusters that are being cleared, unlike the control mock transduced T cells or those wells with Urcat cells. The target cells are then quantified as the area of green fluorescence per image and plotted over time. The graphs show that over 5 days, the CAR-T cells kill the antigen-positive Ramos cells, and the killing event is directly linked to the number of effector cells added to the well. The teal line represents the vehicle-treated tumour cells. With the antigen-negative Urcat cells, there is minimal cell death measured. At the highest ratio tested, we suspect the death measured is from non-antigen-driven killing through the non-transduced population of T cells. So it's possible to combine this live cell assay with flow cytometry readouts to enhance the biological information collected. Alongside Incucyte, Sartorius also supply the iQue Advanced Flow Cytometry Platform, which is a high-throughput flow instrument. As with Incucyte, we offer reagents and kits that can be easily used on the instrument to simplify workflows. The slide details the iQue Human T-cell Killing Kit, which combines phenotyping of T cells using markers for CD3 and CD8, alongside quantification of the activation markers CD25 and PD1. It also allows the measurement of secreted proteins, interferon gamma and granzyme B using iQue beads within the kit. More information around this kit and others that we offer can be found on our website. The graphs show the data from replicate killing plates on day 2, 4 and 7 of the assay. Data from the CD8 positive population are shown in the presence of either antigen positive or antigen negative target cells. The data from this kit demonstrates that only CAR-T cells in the presence of antigen positive Ramos target cells, the black bars in the graphs, display increases in the activation marker CD25, PD1 and interferon gamma. There is also an increase in the granzyme B, indicating cell killing. We're now going to consider the use of CAR-Ts for solid tumour therapy. There is an interest to use the same approach, for example, targeting HER2 for treatment of breast cancer, but early investigations highlighted that adverse effects linked to on-target off-tumour effects. and this is understood to be linked to cells expressing low levels of the target antigen, which were then also attacked by the T cells, leading to serious side effects. To model this potential effect in vitro, we used a co-culture model of a single spheroid of target cells in the presence of immune cells to mimic a solid tumour. For this, we purchased anti-HER2 CAR-T, again with an estimated 50% transduction efficiency for the CAR construct. For the study, we used AU565 cells, shown to be a high HER2 expressor, MBDA-MB231 cells, which in our lab show a minimal level of HER2 expression, and MBDA-MB468 cells, shown to be similar to the isotype control stain cells. And the histograms on the right-hand side of the slide here show the phenotyping data that was collected on the iQue for these cell lines. The cells all express a green fluorescent protein for easy quantification, and a quick version of the protocol is shown at the bottom of the slide here. But basically, the cells are added to the 96-well ultra-low attachment plates for 3 days to allow formation of a single spheroid before immune cells are added to the plate, and all images are captured using the Incucyte spheroid software module. So on this slide, the deep well view shown here is taken at 4 days post-immune cell addition. The green target spheroids can clearly be seen in the plate for all cell types in the bottom row of the plate. In the presence of anti-HER2 CAR-T cells in the top 4 rows of the plate at varying target to effector cell ratios, the AU565s and the MBA-MB231 cells have been eliminated, whilst the MBDA-MB468 cells remain untouched. And no death is seen in the presence of control T cells, and maximal death in the presence of control T cells when activated in a non-specific manner. The green fluorescence can then be quantified over time, as shown in the middle graph, indicating a rapid clearance of the AU565 cells in the presence of increasing numbers of anti-HER2 CAR-T cells. The highest ratio of 2:1, the black line, also shows the fastest killing response. The bar graph quantifies the reduction compared to target cells alone at the 96-hour time point, indicating the lack of response in the MBDA-MB468 cells and maximal killing in the low-expressing MBDA-MB231 cells, as shown in the black bars. I'm going to switch topics slightly now to consider the use of antibodies to drive immune cells to attack tumour cells. And this is being considered using several different approaches. And the first example shown here is the direct conjugation of a cytotoxic payload to an antibody directed to a surface marker or an antibody-drug conjugate or ADC. These work by binding to the epitope of interest, which is then internalized into the specific labeled tumour cell where the cytotoxic payload is released and kills the cells. The next approach is to consider antibody-dependent cellular cytotoxicity, or ADCC, and in this case an antibody is used to label the target cell with a specific marker, and through the Fc gamma receptor interactions of the antibody with an immune cell, the cell is brought into close proximity to the cancer cell, which results in attack of the immune cell and tumour cell death. The final mechanism we're going to consider today is antibody-dependent cellular phagocytosis, or ADCP, and in this example the antibody interacts with a monocyte or macrophage in close proximity to the tumour cells, which results in tumour engulfment and death. And all of these mechanisms can be quantified and tracked using the Incucyte Live-Cell Analysis System. To study antibody-drug conjugates, we're going to utilise the antibody internalisation assay, which works by labelling the antibody of interest with a pH-sensitive dye with the use of our Incucyte FabFlor pH reagent. Once the antibody has bound to the cell and becomes internalised, it enters the lysosomal pathway and into a more acidic environment. This change in pH makes the dye fluoresce, which can be detected on the Incucyte. And the antibody labelling step, as shown on the slide, is very quick and easy and provides a very reliable quantification of this response. In this case study, we used Kadcyla, which is the antibody Herceptin linked to a chemotherapeutic called DM1. It has two mechanisms of attack. First, Herceptin induces ADCC by engaging immune cells to attack the tumour cells, but once bound to the surface, it is internalised and delivers the DM1 directly to the labelled target cells. DM1 then causes the mitotic arrest of the target cells and causing death. The images show HER2-positive AU565 cells after treatment with either Herceptin, Kadcyla, or an isotype control antibody, all complexed with Fab4 pH orange reagent. Both Herceptin and Kadcyla show an increase in fluorescence inside the cell after 24 hours, indicating internalisation, as would be expected as both interact with the same epitope on the cell surface. But at 48 hours, the cells treated with Kadcyla have changed morphology, become rounded, indicating mitotic arrest and an indication of poor cell health. So again, this response can be quantified, and the time course graphs show this quantification, first of the rapid internalisation response indicated by the increase in fluorescence, which has been normalised for the number of cells present in the well. The data demonstrates a very similar potency to both Herceptin and Kadcyla, which is no surprise, as mentioned previously, they interact with the same epitope. We can also analyse the images for cell count using the Incucyte cell-by-cell software module, which enables us to segment individual cells in the image. And we can now see that only treatment with Kadcyla over time results in reduced cell count, indicating target cell death. There is more work we have done with Kadcyla available in, there's more work that we've done with Kadcyla and an additional Herceptin-based antibody drug conjugate, which is available in an application note, which can be downloaded from our website. The final data set I'm going to share is looking at antibody-dependent cellular phagocytosis, or ADCP, where we use an alternative pH-sensitive approach to label cells of interest. For this assay, we use Incucyte Frodo cell labelling kit to label our target cells. These are then mixed with phagocytic immune cells like macrophages and an antibody to drive the interaction. Once the labeled target cell is engulfed and reaches the acidic phagosome, it will fluoresce, as shown in the assay schematic. This assay does require some pre-optimization. You need to ensure that you are only labelling viable cells. You also need to identify an optimal Frodo cell labelling concentration to maximise the assay signal and consider various target to effector cell ratios. So the video shows this assay in action. The well contains bone marrow-derived macrophage, in this instance the effector cell, which are co-cultured with Frodo-labelled Ramos target cells in the presence of a clinical anti-CD20 antibody Truxima. It is important to note that for this assay, the target cells are viable labelled target cells, so we are only measuring ADCP and not efferocytosis of apoptotic cells. The macrophages can be seen as the larger, flatter cells in the image, and if you watch the cell in the yellow ring, you will see the Ramos cells being engulfed and then causing a fluorescence, indicating that the ADCP event has occurred. The graphs show the quantification of this response over time, and the first graph indicates the rapid increase in the ADCP signal, shown as an increase in red fluorescence, with addition of increasing numbers of labelled Ramos cells in the presence of Truxima. The middle graph demonstrates this response can be measured for both Truxima and rituximab, an alternative clinical anti-CD20 antibody, and highlights that as the number of target cells is increased, there is a slight increase in the background seen with the isotype control, but there is still a clear positive signal at all cell densities. The last graph indicates that there is a concentration-response relationship between rituximab and the ADCP response, and this data shown here was captured at 2 hours post-addition, so it's quite a rapid response. Hopefully what I've presented here demonstrates the utility of the live cell analysis for unleashing the power of the immune system to fight cancer. I've exemplified how the various live cell approaches can provide biological insight during the research and development of cell therapies like CAR T cells, and during the development of therapeutic antibodies to engage the immune system by various routes, either direct action with an antibody-drug conjugate molecule or through antibody-directed immune responses like ADCP. I've also introduced the concept of combining live cell approaches with flow cytometry to provide extra context and biological insight. All of these approaches will become more important as the cells or tissues used in the assay become more precious. For more information on all of these products, please check out our website where you can find links to protocols, datasets and multiple documents demonstrating the use of these tools. Just to highlight, we've recently updated the Live Cell Analysis Handbook, now in its 6th edition, which is a great source of information on all our application offerings. Finally, thank you very much for your attendance today.
All right, wonderful. Well, thank you so much, Nicola, for your informative presentation. We will now start the live Q&A portion of the webinar. So if you have a question that you'd like to ask, please do so now. Just click on the Ask a Question box located on the far left of your screen, and we'll answer as many questions as we have time for. Excuse me. All right, let's get started. Our first question here asks, can any of the assays be multiplexed with other readouts?
So yes, many of these assays can be multiplexed. So with label-free cell counting using the cell-by-cell analysis, or with the inclusion of a cell health reagent to assess apoptosis or cytotoxicity. So the user will need to choose the most appropriate reagent and the fluorescence channel to match their instrument and whichever assay it is that they're setting up at the time.
Wonderful, thank you. We've got some great questions coming in from our audience here. This next question asks, are the various monoclonal A/B assays suitable for screening of antibody fragments or nanobodies?
So any of the assays that use the Sartorius FabFlor reagents actually relies on the conjugation to the Fc region of the antibody. So if your antibody actually lacks that entity or that region, obviously they're not going to be able to be labeled with those particular reagents. So both the ADCC and the ADCP assays actually rely on the Fc gamma binding event to actually interact with the immune cells. So again, it's quite important that those regions are present. So if that area is maintained in your antibody, then these assays should all work.
Great, thank you. All right, we have another question here that asks, can the monoclonal A/B driven immune assays be used as part of a drug discovery strategy?
So yes, that's a very key question. So the Incucyte is designed that it can actually assay up to 6 microplates at once. So this can either be a 96 or 384 well plates. So these assays have some optimization, but are suitable for both of those formats. So obviously you can read up to 6 plates at a time. There would be a bit of optimization in there. So the number of wells that you're imaging and the number of channels that you're capturing will affect the time to read, how long it actually takes to read that plate. So therefore it may have some impact on the frequency of the image capture. But yes, they can definitely be, the assays can definitely be integrated into a drug discovery strategy pathway.
Great, thank you, Nicola. Looks like we have time for one more question here, so we'll wrap up after this one. This question asks, is it possible to use organoids in a co-culture setup, for example, immune-driven killing assays?
So our existing solution on the Incucyte is a label-free approach. So at the moment, it's quite hard to look at 2 different cell types in that particular imaging paradigm. So we haven't actually tested that internally as yet, but it's a very obvious next step. And it is one that we get asked about quite a lot.
Wonderful. Well, Nicola, thank you again for your time today and your important research. Before we go, I'd like to thank the audience for joining us today and for their interesting questions. I'd also like to thank our sponsor, Sartorius, for sponsoring today's webinar. Questions we did not have time for today and those submitted during the on-demand period will be addressed by our speaker via the contact information you provided at the time of registration. This webcast can be viewed on demand for 2 years until October 7th, 2025. LabRoots will alert you via email when it's available for replay, so we encourage you to share that email with your colleagues who may have missed today's live event. So until next time, take care, everyone. Goodbye.